Good evening. Welcome. I'm Karen Nelson, Dean of the School of Architecture at the Boston Architectural College, where we have programs in landscape architecture, architecture, interior architecture, and design studies. Tonight, it's my pleasure to introduce the BAC's fourth lecture in the student-sponsored Atelier lecture series of fall 2015, entitled Latitudes. I want to thank the student representatives, Rand Lemley and Luca Townsend, for their help and hard work to make this series possible. Nervous System is a design studio and a science lab founded in 2007 by creative director Jessica Rosenkrantz and chief science officer Jesse Louis-Rosenberg. The duo collaborates very closely while leading a team of 4 in Somerville, Massachusetts. Nervous System works at the intersection of science, art, and technology using processes that employ computer simulation to generate designs and digital fabrication to realize projects. Drawing inspiration from natural phenomena, they write computer programs based on processes and patterns found in nature and use those programs to create distinct and affordable art, jewelry, and housewares. When you encounter the work of Nervous Systems, you seem to be looking at liquid geometries. These designers have played with the sense of compressed and taut lines of force and cut or printed them into being. They have invited plant branching strategies to generate ideas for sconces and for jewelry. Their mode of working allows others to customize and produce inexpensive and highly complex objects. Nervous System intentionally uses manufacturing methods that do not require large facilities or massive manual labor. Their work has been featured in many publications, including The Creators Project, Design Milk, Wired, Dezeen, Design Boom, Fast Company, and Metropolis Magazine. Please give a warm welcome to Jessica Rosenkrantz and Jesse Louis-Rosenberg as they lecture about growing objects.
Thank you, Karen. That was actually one of the best introductions I think we've ever had.
I like liquid geometries. That sounds cool.
And thanks to everyone else at the BAC for inviting us here to talk about our work. So today we're going to talk in depth about 2 projects that we've worked on in the past year. And we'll sort of talk about the ideas and inspirations behind them, our working process, some of the technical details that go into making them a reality.
So as you have been told and may have guessed, Nervous System consists of myself and my partner, Jesse. We started our studio in 2007 when we were both still in school. I was a grad student at the Harvard Design School, and Jesse was an undergrad at MIT. We decided to start our studio because there are a lot of cross-disciplinary experiments we wanted to do, combining our interests that we couldn't really fit into our educational programs. My background is in biology and architecture, and he studied math and computer science. And we wanted to sort of mash up all of these different disciplines and see what happens at the intersection of that.
So our work kind of spans 3 different areas of research that we're really interested in. One is a study of science and nature and really looking at how shape, pattern, and structure emerge from natural processes. And this can span a diverse set of phenomena, be it biological, chemical, physical, geological. We're really interested in patterns where sort of structure emerges from a simple set of rules.
Another area that we spend a lot of time working in is digital fabrication. So thinking about what new opportunities digital fabrication techniques give us for making objects and how they can influence the way we construct the world around ourselves and also express ourselves. And lastly, we're interested in combining these interests in new manufacturing technologies and sort of new simulation technologies into creating new types of design software. So how can we create design tools, design software that's more accessible, powerful, and democratic?
So our media of choice is what we would call code. So we end up making things in lots of different materials, metal, wood, plastics, but really our media is programming, is computer code. And so we don't really traditionally either model stuff in a computer program that is like a CAD package, or we're not really very good at drawing at all. And what we do is sort of write code, which for many people, especially in this kind of discipline, can be very foreign. So we're going to sort of look at a couple of projects as an introduction to that idea. So oftentimes our projects start from a natural inspiration. This is dendritic crystals, which are these branching crystals that grow in agates or limestone. And they can be explained or modeled by this process that scientists have come up with called diffusion limited aggregation, where diffusion of particles is basically particles that move randomly. And then when they hit something, they stick to it. And that is the entirety of the idea. And from that, these complex fractal branching structures emerge. And these ideas of moving randomly and sticking to something can be very easily encoded in a computer program. So when we work, our work sort of looks like this. We have some sort of coding environment. In this case, it's processing, which is an environment made for designers and artists. So it's a good place to start if you're interested in learning to code. And sort of all of this text on the right has encoded this process that's growing the object on the left.
So starting from this inspiration of dendritic crystals, encoding that into a rather simple piece of software that encodes a rather simple process, we then turn that into one of our first jewelry collections that we made in 2007, which is this dendrite necklace series, where every necklace that we make is unique. So far we've made over 4,000 unique necklaces that we've sold. So every time somebody purchases one, they go onto our website and they select the exact growth pattern that interests them, and then we make it. Part of our sort of obsession with digital fabrication is that it can let us make one-of-a-kind things for the same cost as mass-produced items. So if we're going to be using these powerful tools, why use them to make the same design over and over again? Why not open that up towards more customization?
And similarly, it sort of pairs with computational processes where we're, instead of designing a single static thing, what we're designing is a process that generates an infinite variety of forms. And not only do we sort of want to take advantage of that, we also want to express that. We want our work to embody this process and not just be a single static object.
So as you saw in the previous project, a lot of our projects start from a specific natural phenomena that inspires us. In this case, leaf veination inspired a project we did in 2010 called Hyphy. We translated this inspiration into a series of algorithmic systems that we use to grow patterns that are very similar to leaf veination, but differ from them in specific ways that we design. So here you have 4 tweaks on an algorithm we created that grow in slightly different ways to produce similar structures of different types of qualities.
So we might start with an algorithm that was developed in scientific communities or computer graphics to model a specific phenomena. And then we want to take that and sort of explore what is possible within that realm, and not simply to make exactly something that mimics nature or mimics what was originally intended, but sort of explores what is possible within this process.
After creating our initial 2-dimensional explorations, we started to think about taking this algorithm, which was actually originally created by a scientist to explore 2D patterns, and thinking about what that might mean in 3 dimensions. So here is one of our first attempts at that. So we've taken something that was originally intended to be one thing in the scientific community, and then that started to think about what that could mean as a process for design, and specifically more open and 3-dimensional design. And then, similar to the previous project, we ended up translating this into a product. These are Hyphy lamps. Each one is one of a kind. They're 3D printed using selective laser sintering.
So computer-controlled manufacturing techniques like laser cutting and 3D printing open up all of these new possibilities. So we can make things that are extremely complex for the same cost as making things that are as simple as a cube. And you can make things that aren't mass produced, that have infinite variety. There's no tooling, no overhead. So you aren't committed to making thousands of one object. And additionally, they're becoming increasingly accessible everywhere. Schools, libraries, homes have access to these machines, and it's putting manufacturing in the hands of people that never would have had access to it before.
However, everything we just told you is a little bit of a lie. This is the dream of digital fabrication. Its complexity is free and variation is free and it's super easy and everybody can do it. But actually, that's still more of a dream than a reality. There's a big part of the equation missing to make this happen. And for us, that missing part is software. Design software is expensive, it's very difficult to use, and it isn't actually designed to take advantage of the complexity and variation that these machines are capable of manufacturing. Part of the reason is because it's legacy software. It's designed for making things using old manufacturing techniques, not like 3D printing. And nobody has thought about these things before. So a lot of what we do is explore how we can take advantage of those powerful aspects of digital fabrication by creating new types of design experiences. So we create applications like Cell Cycle, which is what you're watching now, which is a web application that sort of leverages simulation and web technologies to make it possible for anyone to co-create their own piece of jewelry. In this case, we're making these somewhat complicated cellular structures fully in real time, so everything you're watching happens just as you're playing with it. It's not a slow process. It's a little physics simulation that you can play around with to design these cellular structures in a customized, playful way.
And so we're trying to make these tools that sort of bring this possibility of complexity and accessibility and variation into the hands of sort of anybody who has a web browser. So the first project that we're going to talk about in depth is called Floriform, which is a project we released earlier this year.
We're going to start with a quote because this is an architecture lecture, and I believe all architecture lectures probably need to have a quote in them. So organic form itself is found, mathematically speaking, to be a function of time. We might call the form of an organism an event in space-time and not merely a configuration in space. This is a quote from Darcy Wentworth Thompson in his book On Growth and Form, which was published in 1917. And the thing that I like about this quote is that it tries to succinctly express the fact that all of the forms that we see in nature, all of the shapes you see in geology and biology and all these other disciplines are the result of complex dynamic processes. And these are processes that are able to grow and adapt to different conditions. And the resultant forms that we see are expressions of those processes and the conditions that generated them. And I'm really into this idea of form as a process. And I think it's a kind of interesting counterpoint to how humans have traditionally constructed objects. We tend to take a very top-down approach where we create objects by directly specifying precise measurements and shapes. Sometimes we almost essentially determine the entire final outcome of a design process in terms of its form at the very beginning of the process, in contrast to these natural phenomena. Computers have been advertised as offering new ways of making, but most design software has tried to very specifically mimic or reproduce the methods by which we've made things before computers. We have software that gives the experience of drafting and painting and sculpting and shipbuilding and model making, all of these things that we do with traditional materials that you work with, like paper and clay and metal. And we've created a really strange digital analog of those tactile experiences in the computer. Our work tries to sort of propose a different direction, so taking inspiration from nature's process-based designs. We're focusing on developing interactive systems that we can engage with. So instead of creating static designs, we're excited about making dynamic processes. Instead of drawing structures, we're interested in growing them.
So this project deals with this idea of differential growth, which sort of goes back in some ways to the sort of very deep idea of the origin of biological form, where if we start with a single cell, how does that grow and differentiate into complex structures? So if a single cell were to just divide and grow uniformly, it would result in, a formless blob. It's sort of like cancer. It just grows uncontrollably. But through carefully coordinated subdivision and differentiation, these very complex structures emerge that have specific reproducible forms and functions. And so it sort of just boils down to the simple idea of, if we grow more in some areas than others, what kind of structure results? And we can divide this into kind of two questions, which is, one is, how does this sort of cellular growth process lead to macroscopic shapes? And then how does this differentiation of growth, how do these differential growth processes get driven? So what drives some area to grow more than another?
And we don't have time to give you the history of all biological theories on this topic. We're trying to give you a little peek into what we find interesting about this, and then we'll sort of back out into design things.
So how does this sort of inhomogeneity occur? And it turns out that in the sort of the whole body plan of an organism is often imprinted very early on in the development of an embryo. And it just starts with a simple gradient that sort of, in this case, in a fly, defines the anterior-posterior axis of the entire organism. So just where's the head and where's the butt? And then from this simple gradient, there's sort of a cascade of reactions that form more gradients and then kind of create this map of the entire organism that sort of tells each cell where it is within the body.
Organisms are also affected by environmental gradients. For instance, in plants, there's a series of very well-documented behaviors called tropisms. Tropisms are directional growth responses to directional stimuli. So they serve essentially just aim a plant towards resources or away from hostile environments. Plants are affected by gradients of light and temperature, humidity, oxygen, nutrient availability, and can also respond to gravity or touch, as in the case of this vine that's growing around a stick.
And so these sort of plant movements are a very well-studied example of differential growth. So by asymmetrically elongating cells on one side of a stem, that stem can bend towards light. And if we were to take this sort of 2D example and move it to 3D, you could have a surface that grows more on one side than the other, leading to positive curvature. This project got started when we came across research by a professor at Harvard named Mahadevan. He was studying the shape of leaves and flowers, specifically looking at sort of the ruffled edge that emerges on these structures. And the theory was that these shapes could emerge through the process of differentially growing more towards their edge. So sort of going back to the simple idea of gradient, we just form a gradient from the edge inward and grow based on that. And sort of the interesting thing about that idea was just that you could have this very simple sort of control mechanism that you could imagine occurring in a plant, and it results in sort of complex shapes, and specifically looked at sort of the blooming of a flower being driven solely by this differential growth.
After we read this paper that had a sort of simple explanation for the emergence of complex forms like this, we started just seeing them all over the place. I'd never actually been that interested in flowers before, but after we read this paper, I started looking at every flower and being like, oh, I could see how the shape of this flower might emerge if it grew more at the edge faster than the rest of the surface. And you can also explain a lot of these ruffly leaf forms, but the more we looked around, the more we started to see structures like this in every branch of life. So you see it in the arms of jellyfish and the shapes of certain types of cacti and anemones and certain types of fungus and all sorts of stuff. One of my favorite examples is this sea slug, which is actually one of my favorite animals in the entire world. It's called the lettuce sea slug, and it has these incredible ruffles on its back. And it actually does a really weird thing where it eats algae, and then it harvests the chloroplast from the algae. That's the part of the algae that does photosynthesis. And then it arrays the stolen chloroplast and all this extra surface area on its back, and it uses it as a mobile solar farm. So it doesn't have to eat. It actually can do photosynthesis from these chloroplasts that it's stolen. It uses all this extra surface area. which is just a totally random side that I just snuck in here because you all need to know about the sea slug because it's the best. Also really excited about this flower. You've probably maybe seen it because it's a pretty popular flower. It's called Celosia cristata. The form on the right is a weird mutation that happens that people like and they call brain flower. The form on the left is the normal form. So it's supposed to look like that. It's supposed to just grow in a cylinder and then it has these flowers that branch out. It looks like a little Christmas tree. But sometimes there's a mutation at the growing point. And instead of growing like a cylinder, it starts to grow like a line. And then the line elongates instead of branching and you get this crazy brain flower, which seems like you could definitely explain that with this model that Mahadevan has in his paper. So we started thinking about, could we create a computer simulation that would explain the difference between these two forms and allow us to explore sort of everything in between this branching Christmas tree and this crazy brain flower? Is there some form that will allow both of these to grow in a system?
So we just started to develop a computer simulation of growing surfaces. And this is sort of the base condition of undifferentiated growth, just growing the same everywhere. And it doesn't simply scale up like you might assume. Instead, it forms these wrinkles all over in kind of a blobby form. So then we started to look at other differential growth modes. So this is inspired by that mutation. We start with a point that then expands into a line. It sort of looks like certain cactuses that have the mutation. This is a point that splits and forms branches. And then this is our original inspiration of growth driven from the edge.
And I guess the thing to think about when you look at these models is all three of these models are grown from the exact same thing. They're all just a hemisphere. And the hemisphere has the same material properties everywhere. There's nothing different about this hemisphere, but when you tell it to grow at different rates in different places, which is what the color is telling you is the rate of growth, it takes on these extremely different typologies of something ruffly versus something branchy. It's just a simple change in rate across the surface. So when we get to this point in our process, we've done something like, we had an inspiration, this weird flower and the sea slug. We wrote a computer program and it like just barely started to work. And we're like, yes, it's working, but where do we go from there? And people often think that when you work in the computer that the results just like pop out and it's magic and you're like not being an artist or a designer because you just wrote some code and then there's something there. But actually there's a lot more that goes into it. And this image for me is sort of standing in for our philosophy as designers who work with code and dynamic systems. I would describe our practice almost as a kind of digital gardening, except for instead of directly cultivating plants, although this project is about plants, so maybe this metaphor is not the best, we're cultivating algorithms. And we're trying to create algorithms or breed systems that have their own innate behaviors, but that we can also sculpt and manipulate. So as we're developing our software, we code sets of influencers, gradients, manipulators, essentially ways of controlling the system that will enable us to sculpt its growth. And what we're going to show you next are some of the controllers that we built into this particular system that allow us to control it through space and time. So in all of these simulations, before red was the rate of growth, but now turquoise is the rate of growth. I don't know why I did that, but just go with it.
All right, so here we're changing the distribution of growth rate through space, still growing from the edge, but sort of changing how quickly growth falls away. These are pretty fast. This is changing the material properties of the surface itself, so its resistance to bending, and sort of increases from left to right. Here we're introducing a directional growth, so instead of growing evenly, it can